Good afternoon. My name is Andre Bastos, and it's a pleasure to join you virtually at CNS in San Francisco 2022. I was really hoping to go in person, and I wasn't able to make it this year, but I'm really looking forward to seeing many of you in person as things start opening up again. And with that, I'll go ahead and start my talk. It's titled A Spectro-Laminar Framework for Cortical Computations. And so the overview of the talk is that I'm going to talk first about the predictive coding model, which, as many of you know, is now one of the dominant models in cognitive neuroscience, and how we've attempted to test it with multilaminar electrodes. I'll then talk about a ubiquitous cortical dynamics involving superficial and deep layers, which might be involved in this process, and test the model causally with propofol anesthesia in the final section. But for now, let's launch into what does the predictive coding model actually say? So predictive coding basically says that there's a hierarchy of areas in the brain, and top-down connections from higher order to lower order cortex are involved in carrying predictions about what you're going to see next. And the sensory inputs, they arrive, and when the sensory inputs match the predictions, then nothing needs to happen. And so this is a great way to save energy for the brain. But when there's a mismatch, then a prediction error computation takes place and is sent feed forward up the network. And so the hypothesized circuit is that these prediction errors occur in layer 2, 3, and in pyramidal cells in layer 2, 3, superficial layers, and are relayed up the hierarchy. So we set out to test this model with the following experiment. We trained monkeys on a visual working memory task. Monkeys fixated, they were shown a sample stimulus, which they held in mind over the delay period, and then performed a saccade to the previously sampled item identity. And then we trained monkeys to perform this task in one of two ways. Either they were shown the same sample over and over again for many trials in a row, A, meaning the car sample many times, or in an unpredictable setting. And so we manipulated sample predictability, and we recorded with these 16-channel or 32-channel multilaminar electrodes in a network of prefrontal, parietal, and visual areas, trying to go perpendicular to the cortex in order to be able to then estimate neural activity in both superficial and deep layers. What we found is that across the network, there was more spiking to an unpredictable stimulus than compared to a predictable stimulus. And in addition, there was more gamma to an unpredictable stimulus. So on the y-axis is LFP power change. Positive values indicate more LFP power during an unpredictable cue. Negative values mean more LFP power to a predictable cue. And this was a power analysis done in the sample period. So in the sample period, as you can see, there was more gamma across cortex and also more theta across cortex, with one exception, and that makes sense. Gamma and theta tend to go together. But there was the opposite pattern in the alpha beta range, where there was more power to predictable stimuli, and this alpha beta band changed slightly across areas, but it was always there in each individual area. We then tested for the laminar differences in these spiking and LFP effects. What's shown here is now the multi-unit analysis, so the spiking activity, the difference between an unpredictable sample and a predictable sample, but now during the sample period, but now broken out into superficial layers in orange and deep layers in blue. And in V4, there was indeed more superficial layers spiking to an unpredictable sample. And there was also more gamma power to an unpredictable sample, again, with a preference for superficial layers in both V4 and PFC. Now, in addition to that, we also did an analysis where we looked at the selectivity, whether the neuronal selectivity modulated this effect. So we separately analyzed neurons that preferred the sample that was being predicted compared to neurons that did not prefer the sample that was being predicted. The idea here being, does prediction reach down to the appropriate neural ensembles that need to be enhanced or inhibited? And the answer is yes. So superficial layers displayed more of a enhanced gamma to the unpredictable versus predictable sample when it was the preferred sample. And in the deep layers, there was a similar effect seen, but now the opposite effect, more predictable, the more predictable the sample stimulus, the more beta, but only for columns that corresponded to neurons that had selectivity for the sample stimulus. So these observations led us to propose that predictive coding may be implemented via predictive routing. So when a prediction matches a stimulus, meaning that you have a top-down prediction, you're predicting that you're going to see sample stimulus A, and you're shown sample stimulus A, then what happens at the level of cortical processing in the visual cortex, for example, where we recorded? A host of mechanisms appears to be in place. Namely, there's top-down data from higher order cortex, and this actively prepares the specific cortical columns to receive this predicted stimulus by enhancing the alpha beta. And this top-down signal originates in deep layers and targets deep layers in a specific way. This enhanced deep layer beta in turn inhibits superficial layer gamma and superficial layer spiking. And we've shown this in a number of manuscripts and papers. And as a result of this, of all of these inhibitory mechanisms that predictions put into place, there is less feed-forward output and there's less gamma. And so the stimulus that's arriving to a prepared cortex is not processed very deeply and does not evoke a lot of activity. Now, in the case of a prediction that does not match the stimulus, we're predicting now B to see B, but we're actually shown A, then basically what we're saying is that there is a failure in all of these inhibitory mechanisms. So the stimulus A is arriving at a relatively unprepared location in cortex. So there's less alpha beta top down, there's less inhibition of superficial layers, and as a result, the sensory sample evokes a lot of activity. And we call this activity a prediction error. So there's more superficial layers spiking in gamma feed-forward output. And so the main difference here that we are proposing with predictive routing is that the real heavy lifting, the real computational work is being done by these dynamics, which we see over and over again in all of these different tasks. Wherever we measure throughout cortex, we see these interplay between alpha, beta, and gamma. And with that, I'd like now to launch into the second part of my talk, seeing whether this superficial versus deep layer dynamic, how ubiquitous is this across cortex? One of the first observations I made during my postdoc with Earl Miller when I was at MIT is the following. When we place probes perpendicular to cortex, on the y-axis is the location of the laminar depth of the electrophysiological recording, in this case, local field potential, with 0 being the approximate location of layer 4, superficial layers 2 and 3 up here, deep layers 5 and 6 down there. And frequency is on the x-axis, and the color corresponds to the relative power of each frequency, which layer had the most relative power. So this really jumped out at us when I first started doing these laminar recordings. And what we noticed was that there was a preponderance for gamma in the superficial layers and a preponderance of alpha beta in the deep layers. So then with other collaborators, including Diego Mendoza-Halliday, Bob Desimone, as well as Noah Lee, we sought to then expand this to other areas. So what's being shown here are those same plots, but now more relative power in yellow and less relative power in blue, across a completely different set of areas, MT, MST, LIP, PFC. Here are grand averages across individual monkeys, individual probe. Wherever we look in the cortex, here's the studies that I've done before, parietal area 7A and PFC, which I showed in the previous slide. This pattern kept popping up again and again and again. So it's a pattern that's present in individual probes, different brain areas, different monkeys, and different studies. And most recently, I came to Vanderbilt University to start up my lab, and in collaboration with Alex Meyer and his very talented PhD students, Brock Carlson and Blake Mitchell, we've now observed the same pattern also in V1, namely the superficial gamma and deep layer alpha beta. And now, to be fair, this pattern had already been discovered and seen by Alex Meyer and others in V1. And what we're doing here very specifically now is quantifying the degree of overlap. And what we've been able to show is that no matter where we look in the brain, if we take this swoosh pattern from one area, this gamma superficial alpha beta deep, that tends to predict the spectro-laminar swoosh pattern in all of the other areas. And in addition, going beyond just the superficial versus deep layer distinction, what we were also really interested in is determining the specific anatomical layers that express these oscillations. And so here I'm showing pioneering work from Charlie Schroeder and colleagues, wherein he performed current source density analysis in different visual areas and performed tiny electrolytic lesions to then look at the exact layers that were corresponding to different CSD sync and source patterns. As pioneering as this work was, it didn't quantify the exact layers where rhythms were coming from. And it also relied on this current source density method, which necessitates that you have a strong excitatory stimulus. And it's not clear how to use this method for non-sensory areas, like prefrontal cortex, which still have very beautiful lamination, as I'll show in a minute. And so in collaboration with John Kass in his lab, we've developed a histology processing pipeline to address these questions by looking at the electrophysiological laminar rhythmic pattern of gamma and beta, performing tiny electrolytic lesions, and then histology work to map specific layers to electrode sites. This is work done by a very talented undergrad in my lab, Max Lichtenfeld, and another talented research assistant in my lab, Patrick Meng. And what we did here was we performed these tiny electrolytic lesions. These are lesions that show up only about 50 to 100 microns. In this case, with the multilaminar probe, we stimulated in these two red contacts, which generate these small orbs over here. This is another site on another electrode, which we've also mapped, but it's not this example that I'm showing. And then now that we have this data in hand, we can then reconstruct the exact probe location and map each electrode location to a specific layer. So here's layer 1, layer 2, 3, 4, 5, and 6. And moreover, we can take the corresponding and simultaneously recorded electrophysiology and show exactly where the gamma peak is in relation to the layer. So here's the relative power of gamma shown in red, relative power in the x-axis, depth on the probe in y-axis, and the relative power of alpha beta is shown in blue. And now we can exactly take the peak and parameterize that to the layer. In this case, layer 4 for the gamma peak, layer 6 for the beta peak, and the gamma beta crossover, the point where the relative power crosses over in layer 5. We then repeated this for many different examples in 2 different monkeys, and we're now processing data from a third monkey in other areas. And in general, what this shows is that in red, you see the location of the peak gamma power. It tends to be in layer 2, 3. In blue, the peak beta power tends to be in layer 5, 6. And in purple, the crossover point, which tends to be around layer 4. And now we can quantify across an entire population where we can really be specific about where is the mean of the gamma peak, where is the mean of the beta peak, and where is the mean of the gamma beta cross, which layers are those things tending towards. So on the y-axis is the distance from layer 4, where each peak on different probes, a total of 10 probes, was observed. The gamma peak maps nicely onto the border of layer 2, 3. The beta peak maps nicely onto the border of layer 5, 6, and the gamma beta cross to layer 4. And so what we think is going on here, this is just one possible model that we're now pursuing. We're pursuing other hypotheses as well, but this is one of the models, is that we have, of course, recurrent connections between excitatory and inhibitory cells, and different time constants of inhibition may generate gamma in superficial layers, but alpha beta in the deep layers. And again, this was shown not only in PFC, but we're now expanding this observation to other areas as well. And lastly, now I'd like to turn to a causal test of the predictive routing model that we did with propofol anesthesia. And this was also work that I began during my time at MIT as a postdoc, with Earl Miller, and collaborating with Emery Brown and Jake Donahue. And what we did was we performed electrophysiology recordings of both LFP, shown in these upper traces, and spiking across a network of frontal, parietal, and sensory areas, and now in auditory cortex, area STG, also referred to as TPT. We performed these electrophysiological recordings, and we gave the animals propofol so that we can track exactly the change in the conscious state and the change in rhythmic dynamics. And we did this for 2 reasons. First, we wanted to understand better medically how it is that anesthetics work to render patients unconscious, for example, during surgeries or medical procedures. But secondly, and also very interestingly, it turns out that propofol profoundly remodels these brain dynamics, imposing a very slow frequency wave in the LFP and largely eliminating beta synchrony. And these slow frequency waves, they synchronize spiking across cortex. So spiking gets less numerous, but it also gets really locked to these different waves of the LFP. And so propofol-induced unconsciousness is accompanied by a host of factors. These strong, slow waves create alternating on and off states for spiking, reductions in spiking activity throughout the network, with the strongest reductions in spiking happening in prefrontal cortex, and also reductions in cross-area beta, with a relative sparing of sensory evoked activity in early areas. And so with all of those things in mind, we thought it would be a good, an interesting task to show the monkeys a predictable sensory stream or unpredictable sensory stream. And again, to reiterate the predictive routing model, we have these superficial versus deep layers with feedback coming from deep layers in alpha beta, providing the prediction, the difference between the prediction and the sensory state being linked to gamma and superficial layers, which is feeding forward. And then specifically with propofol anesthesia, that has a really profound effect everywhere on the brain, but it affects higher order prefrontal cortex the most and knocks out the alpha beta feedback while sparing some gamma and spiking responses to sensory stimuli. So we wondered what would then happen when we altered the network in this way during a task which would evoke some kind of predictable versus unpredictable response. And for this, we use a sensory prediction paradigm that involved both local oddballs, which were violation to the local sensory stream. So in this case, auditory tones, beep, 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 boop. The boop would be therefore a local oddball, as well as global oddballs, which are violating the more generalized pattern that is seen across several of these tone sequences. This paradigm was adapted from Stan Dehaene's lab and was published in many studies showing that you can evoke robust neural responses to these, especially to these local oddballs. To then, again, tie that to the predictive routing model, we would expect that local oddballs would be involved, would evoke feed-forward gamma prediction errors, whereas predictable stimuli would evoke feedback predictions. So we, again, recorded from these different areas, including auditory cortex and FEF, which I'll focus on today, and administered propofol while also recording an awake baseline state as well as a recovery state. And so now this analysis was done by a very talented PhD student in my lab, Sophie Chang, who happens to be at CNS. She's also going into much further details on this topic at her poster, which you can check out in session E. It's poster number 59 on Monday. And what she found is that when she now calculated the power differences between an unpredictable stimulus versus a predictable stimulus. So for example, a local oddball beep, 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 boop versus beep, 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 beep. We now time lock the time frequency analysis to the onset of the local oddball, which is here at time 0. On the x-axis is the time after the oddball. On the y-axis is the frequency of the LFP response. And in color, red colors correspond to more power at those frequencies for an unpredicted stimulus. Blue colors, more power for a predicted stimulus. And so, not surprisingly, she was able to reproduce what's known from prior work. There's more gamma to an unpredictable stimulus, but more beta to a predictable stimulus. But now what happens during anesthesia in this sensory area TPT that we recorded from? And what happens there is that there is an overall increase in power. It still is in the gamma range, but the alpha beta reduction, or in other words, the alpha beta enhancement during prediction is completely wiped out, which we think makes a lot of sense. If you have less of this inhibitory alpha beta mechanism, then you have a preponderance to express more gamma. And so now to conclude overall the talk, we propose that predictive coding is implemented via predictive routing. And predictive routing says that you prepare the cortex through alpha beta inhibition. And then when that prepared cortex actually gets its stimulus, then it doesn't evoke very much processing. But in the absence of that preparation, in the absence of that beta inhibition, you have enhanced feed-forward gamma and you have enhanced spiking. And the spectro-laminar motif of different layers having these different oscillations appears to be ubiquitous across cortex. We're now testing whether it's also ubiquitous across different species. And this LFP beta modulation to oddballs or unpredictable tones is lost during propofol-induced unconsciousness. And without this beta inhibition, the LFP gamma modulation to an unpredictable input is enhanced, which supports the predictive routing model. And so with that, I'd like to thank the many collaborators and contributors to this work, many of whom I've mentioned during the talk, and my great lab at Vanderbilt with Michaela Maddox, Sophie Shong, Patrick Meng, and Max Lichtenfeld, my collaborators from the Alex Meyer lab at Vanderbilt, Alex Meyer, Blake Mitchell, Brock Carlson, and Jacob Westerberg, collaborators on the histology from John Kass's lab, and collaborators at MIT, where I began this work, my postdoctoral mentor, Earl Miller, Robert Desmond, Diego Mendoza Halliday, Noah Lee. And I would like to thank my financial support as well. And lastly, if you're interested in this topic, please get in touch with me. I'm just opening up the lab now. About a year ago, we got started. And so if you're interested in these topics of cognition, computation, neural computation, and consciousness, please join us at the Bastos Lab and please get in touch with us. And with that, I'd like to thank you very much for your time.